Okay, so my genetics project was over progeria. Progeria, or commonly known as Hutchinson-Gilford progeria syndrome, actually causes accelerated aging in children, which they recognize about a year after birth. So the children actually appear phenotypically normal at birth, and then about a year after that, they start developing hair loss, wrinkled skin, enlarged head, a small jaw, thin lips, a beak nose, and visible veins throughout their face and their forehead. And these are all signs of severe aging. So if they're aging 8 times as normal as a regular person, their average lifespan is about 14 years old. So they're not reaching sexual maturity, which means that there is no genetic transmission from an affected individual to offspring. And we see that because it's a de novo heterozygous single base substitution mutation, which means that de novo means that it's a random mutation that has 3 places of occurrence. It can occur in the father's sperm, the mother's egg, or in the fertilized zygote. And it has a chance about 1 in 8 million. So it's really a rare thing to happen. And there's only about 40 children in the world that actually have the progeria. Okay, and what it actually does is this mutation produces, in a normal individual, it will produce a pre-lamin A. And then this will undergo post-translational modifications to become mature lamin A. And mature lamin A is just a nuclear lamina, nuclear lamin that will form the nuclear lamina, which is the meshwork that gives the nucleus and the nuclear membrane structure and strength. So we see here where we have a normal nucleus that has a normal lamin A, lamin B, lamin C. And then over here, we see a nucleus that has the mutated lamin, which is actually going to form a protein known as progerin. Now, progerin does not function as the same as lamin A, and that leads to several problems affecting the nucleus, cell division, and transcription of some proteins. And it will actually cause the nucleus to not be a smooth, a round circle. It's going to actually cause it to be wrinkled, and that's going to make it lose some of its integrity and some of its function. And that will lead to all these signs and symptoms and ultimately lead to the death of the affected individual about the age of 14 due to either heart attack or stroke, most commonly, as a direct implication of having progeria. So the mutation will produce this pre-lamin A, and then it will undergo several post-translation modifications. So the first one is going to be pre-lamin A is going to be added a farnesyl group. Okay, this right here is a farnesyl group. It's made of isoprene units, and it's actually going to be added onto the end on these 4 amino acid residues. And I want you to notice right here, we see this blue line. The blue line represents 50 amino acids. So when the gene, the mutated gene produces, it produces one with missing 50 amino acids, which is a 50 amino acid deletion. So that's going to cause all the rest of the post-translational modifications to be errors. But in the normal one, it has the 50 amino acids. And I say it's critical because it's critical to the protein function at the end. So the farnesyl group is transferred in the first step of the post-translational modifications, and then an enzyme named endopeptidase will actually come and cleave off these last 3 amino acids. Now that's going to expose a carboxylic acid that has a negative on one of the oxygen atoms, and to get rid of the negative, we need to add a methyl group to it. So methyltransferase enzyme is actually going to do that. So it's actually going to add this methyl group right here that we see, and then this guy is going to go to the nucleus. So this carboxyl methylated group here and the farnesyl group are actually, they serve as a nuclear localization type of signal. That's actually going to give the molecule, it's going to give us a structure and the function, and it's allows going to actually embed into the nuclear membrane using this. But once it gets in the nucleus and it begins doing its job, it doesn't need this anymore. So this enzyme ZMPSTE24 is actually going to cleave it in half. And we see here it cleaves it where we have blue here and blue here. So it's actually going to cleave it and this is going to be a signal as to where to cleave. So, and that'll be important when we look over here. But this will actually cleave it in two. We'll get rid of the farnesyl and carboxymethylated group, and we will only have this lamin right here. Okay, and this lamin will combine with the other lamins, lamin A, B, and C, to actually do their job, form the nuclear lamina, and give us a nice nucleus that has proper function. But if you have Hutchinson-Gilford progeria syndrome, you have a pre-progerian protein produced instead of pre-lamin A from the mutated gene. So pre-progerian is produced, and pre-progerian has a 50 amino acid deletion, and we see here that the blue represents 50 amino acids. That's in the normal, and it's not in progeria, and that's going to be significant. So the farnesyl group is added, and endopeptidase cleaves off the last 3 amino acid residues, and then the methyl group is transferred. But once we get in the nucleus, our ZMPSTE24 enzyme, it doesn't know where to cleave because it doesn't see this group right here. Okay, we don't see that group here. So if it can't recognize it, it's not going to cleave it, and that means we are stuck with this long thing with the farnesyl group and the carboxymethylated group on it. And now this group right here is going to inhibit its function and cause it to not do its job correctly. And that's going to lead directly to our nucleus being misformed, misaligned, and it's not going to lead to proper nuclear functions. And I think that's it.